My name is Savannah Sadler, and I will be talking to you about today the receptor tyrosine kinases and its relationship to cancer. So our objectives for this lesson, after this lesson, I will have learned what a receptor tyrosine kinase is, the mechanism of activation and signaling of RTK, and how receptor tyrosine kinases are related to the development of certain cancers. So what is a receptor tyrosine kinase? RTK has a role in the cellular process, including growth, mortality, differentiation, and metabolism of cells. Each RTK contains an extracellular ligand, binding domain, a single transmembrane, helix membrane, and a carboxyl group. RTKs are activated when growth factor ligands bond to the extracellular regions of the RTKs, and the receptor is activated by the ligand-induced dimerization and/or oligomerization. So the mechanisms of action and signaling. So tyrosine phosphorylation is very important to inter and intracellular communication. The RTK family contains epidermal growth factor receptor, or EGFR, platelet-derived growth factor receptor, fibroblast growth receptor, also known as FGFR, and vascular endothelial growth receptors. Defining specifically the hepatocyte growth scatter factor, or HGF/SF, ephrin receptors and insulin receptors are also included. RTK is important in cellular signaling pathways and are active in the embryonic development and adult homeostasis. So ligand-induced dimerization of RTK in extracellular regions. Receptor dimerization is completely ligand-mediated without any direct contact between the extracellular region of the two receptors, such as TRKA, the NGF receptor. The second way is dimerization is completely receptor-mediated without any physical interaction between the two active ligands, for example, ERBB family. The third way is ligand homodimers bind to receptor molecules that interact with each other across the dimer interface, such as KIT and SCF receptors. And then lastly, a combination of the ligand bonding and direct receptor-to-receptor contact. It also participates in receptor dimerization. In some cases, the dimer or higher order oligomer forms of RTK can activate ligands on the ERB family. The ERBB family is important to the cause of cancer. The receiver kinase is formed when the C lobe of one kinase domain in the dimer pair comes into physical contact with the N lobe of another kinase domain in the dimer pair. So RTK's role in cancer. So RTK plays a large role with growth factor receptors. It has been observed that RTK is responsible for a various number of cancers. Cancer is caused by the function mutations or receptor ligands being overexpressed. Diseases such as cancer are formed when the RTKs are not regulated properly. There are 4 different mechanisms that can cause abnormalities: gain-of-gain function, genomic amplifications, chromosomal rearrangement, and autocrine activation. So activation by gene of function mutation is the first one from the previous slide. So when RTKs lead to downstream signal transduction it does not go through the checks and balances that it typically would go through. This may cause driver mutations, which interfere with cell proliferation. The three other areas that occur are the extracellular domain, ECD, the transmembrane domain, TMD, and the juxtamembrane domain, JMD. Mutations can lead to the overexpression of EGFR proteins that produce phosphorylation. Mutations in the ECD leading to cervical cancer caused by the abnormalities in the cysteine residues allowed for abnormal dimerization through intramolecular disulfide bonding. In thyroid contact, it is seen to be rearranged during infection. Mutations also cause KIT in gastrointestinal stromal tumors. And lastly, mutations with the TMF can cause non-small cell lung cancer. So, Lee-Fraumeni syndrome is a common syndrome that has to do with TK. So, Lee-Fraumeni syndrome is an inherited condition that puts you at great risk to develop certain cancers. While any cancers can develop, the most common cancers are sarcomas, including muscle, bone, and connective tissue cancers, breast cancer, leukemia, and adrenocortical carcinoma. The TP53 is a protein that is responsible for tumor protein mutations, and it can lead to cell proliferation. So overexpression of genomic amplification. The overexpression of RTK has been shown to be a factor in EGFR and GMB lung, esophageal, and thyroid cancers. The overexpression of HER2/ERBB has been found in lung, bladder, breast, and gastric cancers, and the overexpression of methionine has been seen in lung and gastric cancers. Gene amplification can be influenced by common chromosomal fragile sites, defects in DNA replication, and telomere dysfunction. Amplification of EGFR, ERBB2, MET, and FGFR1 are found in the lung and breast cancer. Another example could be the overexpression of FGFR3, which has been seen in breast and bladder cancers. So the third one is chromosomal arrangement. So imatinib is the first tyrosine kinase inhibitor to be approved by the FDA. Its function is to target all ABL kinases. It has also been a treatment for patients with chronic myelogenous leukemia. So some of the stats what ALK is prevalent in different cancers: 3 to 7% in non-small cell lung cancer, about 10% in spitzoid neoplasms, and then trace amounts were found in colon and thyroid cancers. The fusion of oncoproteins, for example, TRKA, TRKB, and TRKC, tyrosine kinases, have identified 9 tumor types, including sarcoma, melanoma, thyroid, lung, colon, breast, head, and neck cancers. The first feature of fusion includes a regulatory unit to dictate the expression of fusion. The second feature is that most partners wish to contribute to is an oligomerization domain to protein and ligand-independent kinases. Lastly, most commonly, oligomerization domains are found in fusion partners are called coiled coil domains. Lastly, we have autocrine activation. So autocrine refers to the situation in which cells are secreting cells themselves. Autocrine plays a large role in the body. Vital autocrine activation can lead to tumor formation. Autocrine activation RTKs are found in several different cancers. For example, TGF alpha and EGFR, or HGF and MET, or SCF and KIT autocrine loops. RTK autocrine loops can work with other autocrine growth pathways, which will result in tumor development. So lastly, for our conclusion, after this lesson, you should now know what a receptor tyrosine kinase is. We discussed that it is a role of cellular processes, including growth, mortality, differentiation, and metabolism of cells. Our second objective, the mechanisms of activation and signaling of RTK. So it allows for phosphorylation, contains several growth factors, and is essential for embryonic development and adult homeostasis. And then lastly, how receptor tyrosine kinases are related to the development of certain cancers. Since RTKs are responsible for cell proliferation and death, they can be mutated or overexpressed, causing tumors and cancers. And then lastly, I have my reference page. Thank you.